All right, so that last video was obnoxiously long, so we're going to try to fly through bacteria and archaea, but this is definitely one that or chapter you're going to want to look at for any species or any clades or phyla that you have in those categories, and you all should have at least one in each of those categories. So this is just giving you an overview of a visible sign of prokaryotes, that the Great Salt Lake gets its pink color from them. Prokaryotes can live pretty much anywhere you can possibly think of, even places where other living things would never even begin to try to live. Um, they may be really tiny, but there are lots of them out there, and we have two domains in which we find them in, the bacteria and archaea. We've talked about this before, but prokaryotes were probably the first living things on our planet. We can find them as unicellular. Sometimes they are very happy to work in concert with one another, so they will colonize, form colonies with one another. They're very, very small, and the three most common shapes that we tend to see them in are spherical, rod shape, or spiral. Their cell wall is one of the key structures that allows them to survive. We talked about that a little bit, cell walls, when we were covering organelles earlier on this year. Um, we have eukaryotes that can have cell walls, your plants and your fungi. They're made of cellulose or chitin. But bacteria cell walls are often made from peptidoglycan, which is sugar polymers that are linked with polypeptides with proteins. Archaea cell walls will have the polysaccharides, and they will have proteins, but they don't tend to have the peptidoglycan. And so a gram stain can be used to distinguish between bacteria based on their cell walls. And I'll show you a picture that more fully shows this, that more clearly shows this. Gram-positive bacteria will have lots of peptidoglycan in them, and those are the ones that antibiotics are able to help to get rid of. While gram-negative bacteria don't have as much peptidoglycan, and they are more resistant to antibiotics as a result. So there's an example. The one on the left is the gram-positive. The one on the right is the gram-negative. Again, that peptidoglycan layer, which is on the outside of the bacteria, can be neutralized depending on the type of bacteria it is by antibiotics, and it will cause the gram stain to turn purple, while gram-negative bacteria will not turn purple. They'll stay that red because the carbohydrates on the outside prevent the dye from being taken up, kind of like the antibiotics aren't able to work as effectively. So some structures that we tend to see in the prokaryotes are capsules. That just is an extra layer. It helps them to stick to things, and it does help to provide them with some protection. They can have fimbriae, which will allow them to stick to other parts of their colony, other members of their colony, or to whatever they are trying to bind to. And then they can have pili, which will allow them to exchange DNA and allows them to have the rapid mutations. So, motility, they can move towards or move away from a stimulus. They exhibit taxis. They can also do this with chemicals, and they often are moving by flagella. Sometimes they'll just have them in one side of the prokaryotic cell, or they can have them on both sides, or they can be scattered throughout. Um, Flagella are not able to be used as a way to distinguish between, sorry, flagella are not in common between bacteria and archaea and eukaryotes, so that they all can have them. They are not the same composition. Okay, so there's just an example of a flagella. And so the proteins that are used in the bacterial flagella also do other things in the bacteria. So this is most likely a trait that has evolved as the bacteria were wanting to be able to do other things. So it says this is an example of exaptation, or structures that are present take on new functions. So if this was used initially as a way to remove things from the prokaryotic cells, um, it was able to adapt and take on an additional function that the prokaryotic cells needed. Again, they don't have nuclear membranes, they don't have a nucleus. Their DNA tends to be circular, and it's just located in a general region, the nucleoid. They can have plasmids that are present as well, smaller rings of DNA. And those plasmids, again, can move between cells. And we've talked about the replication and transcription and translation before. Their ribosomes are a little bit smaller. And because these processes aren't done exactly the same as they are in eukaryotic cells, that's what allows us to take antibiotics and not typically, although depending on the antibiotics, you can have other side effects, but you typically would not harm yourself by taking an antibiotic to treat a bacterial infection. Uh, Prokaryotes replicate by binary fission, and they can do it quite rapidly. Because of how small they are, because they have a smaller genome, it makes it a little bit easier. Um, if they are in an environment where they are unlikely to survive, they can form endospores, where basically they take their cell and they divide it in half, and they basically go inactive for a period of time. They can be that way for centuries, which is part of the reason when you take an antibiotic, you need to take it all the way through and make sure you've killed off all those bacterial cells that you have present. You don't just take it for until you feel better. Because they're able to reproduce so quickly, that is part of why they are able to evolve and adapt to their environments. Um, so even though they might be small, they can do lots of things. Okay, so there's an example of an endospore. Um, how prokaryotes are able to have such a range of genetic variation and genetic diversity. Again, they reproduce pretty rapidly, which can cause changes in the DNA. They can have mutations occur, and then they can have the genetic recombinations where pieces of DNA are transmitted from one cell to another. Um, in general, because they were reproduced by binary fission, they are identical. Um, if there are mutation rates during that process, and they continue to rapidly reproduce, those mutations would be passed on fairly quickly. So that is one factor that influences their variation. Um, they can take in DNA from other sources, either through transformation, we did that in our lab, transduction, and conjugation. And when these genes are moving between different species, we call that horizontal gene transfer. When you take up DNA from your surrounding environment from something that is foreign, that's called transformation. Transduction is when we're taking in genes, when bacterial cells are taking in genes from viruses. Okay, and there's just an example. If you look a little closer, you can see how the donor cell started as A plus B plus, and then it ended up as A plus B minus by taking in that DNA and recombining it as part of its genome. Conjugation is when we're having a transfer of material. It only can go in one direction. You have to have that F factor to be able to make the pili that allows the DNA to be transferred. If you don't have the F factor, you can accept the DNA, but you are not as likely to donate your DNA. You can transfer that F factor gene as a result of conjugation. And when you take in that DNA, you no longer are going to be the same, or no longer have the same genome that you had previously. Okay, so again, in this particular one, we have an F plus cell, which is a donor, and an F minus cell is a recipient. And you can see as the DNA is moving through the pili that now we have made two F plus cells. Okay, and here you're just seeing where part of the chromosome is being transferred. So we talked about F plasmids. We're also going to talk about R plasmids, and those are the ones that can have genes that will help to resist antibiotics. So if you have that being transferred, that can help to protect the bacterial cell and make it less susceptible to antibiotics. And that's part of why doctors are definitely a lot more cautious with giving you antibiotics if you have a viral infection, because the bacteria can build up resistance and then they won't be able to work when you really need them. Prokaryotes um, are categories in many different ways. And we're going to talk about these as well with the other clades and phyla that you're going to be exploring. They can get their energy from light when they're phototrophs. They can get their energy from chemicals when they're chemotrophs. They can get their carbon source through carbon dioxide if they're an autotroph, and they can get their carbon source through organic material when they're a heterotroph. And then we can combine these together to get different forms of nutrition. We can have photoautotrophs, we can have chemoautotrophs, we can have photoheterotrophs, and we can have chemoheterotrophs. Okay. And then there's just some examples of different species. And you can see with these nutritional modes, this definitely might be a chart that will benefit you with your project. You can see where it's got not just the prokaryotes on there, but it's also got protists, it's got plants, it's got fungi, it's got animals. Okay. um, how oxygen plays a role in being able to metabolize things. Um, we are obligate aerobes, we being humans, because we require oxygen for cellular respiration to take place. There are some living things that will not be able to survive if oxygen is present. They are obligate anaerobes, and they use fermentation or anaerobic respiration to be able to metabolize what they need. And then we have facultative anaerobes, which can both live in the presence of oxygen and without oxygen. We're not going to spend a whole lot of time talking about nitrogen metabolism, but nitrogen is also a way to be able, you need nitrogen in your amino acids, you need it in your nucleic acids, and prokaryotes are able to fix nitrogen to be able to take some of that nitrogen and use it to make their proteins and their nucleotides. Um, Sometimes prokaryotes will cooperate with one another so that they can utilize what they have present in the environment when they wouldn't be able to do it solo. And there's just an example of Cyanobacterium anabina. It has photosynthetic cells and nitrogen fixing cells that are called heterocysts. So the individual cells are able to exchange products, and so they're able to work in concert with one another. We also see this with biofilms. So now that we are able to analyze more genomes of different species of prokaryotes, this is what has led us to, back when I first taught biology, into splitting the Monera kingdom into bacteria and archaea, and has now led to the domains. And again, we're going to keep working on this, especially as we're able to sequence more and more genomes and look at the gene products that are formed from those different phyla and clades of bacteria and archaea. So we'll talk about eukaryotes much more in future chapters. This one we're going to be focusing on the bottom two, the different groupings of archaea and the different groupings of bacteria. Remember that they are not kingdoms in either of those or phyla necessarily. They just tend to refer to them as groups. It does on the kingdom and phylum classifications and everything that comes under that will fall under eukarya. Okay, so we're going to start with archaea. Um, Archaea and bacteria, the biggest difference between them is the environments that the archaea are able to live in. They tend to be found in much more extreme environments. We have halophiles and we have thermophiles. Halophiles, like saline, halophiles are halogens. Saline means chlorine. So chlorine is a halogen. Sorry, I had to get some chemistry in there. And then thermophiles with the heat. Um, This is another chart that will kind of allow you to compare the bacteria to the archaea to the eukarya. I think I talked a little bit about cell walls on your project. I don't believe any of these others are really going to be super needed for your project, but that just is a kind of a comparison about how they vary between the different domains. Methanogens are going to produce methane as a waste product. They are very strictly anaerobic. If oxygen is present, they're going to die off. Um, and it's thought that methanogens might help us get a better idea as to the different types of groups of archaea that are out there and how everything has evolved over time. Um, Bacteria are going to be the prokaryotes you're more familiar with, and there's lots of different nutritional needs among those groups of bacteria. The 5 groups of them are listed there. I believe all of you have at least one of those groups, and one of you has 2 of them. All right, so proteobacteria are gram-negative bacteria, so they can't take up that stain, and they are photoautotrophic, chemoautotrophic, and heterotrophic. Some are anaerobic, some are aerobic. And there's some different examples for you. E. coli is a very common one that causes bacterial infections. You may have heard of Legionella before and Salmonella. Vibrio cholera. Helicobacter pylori is definitely a source I've been told with intestinal issues. Chlamydias are parasites that can live within animal cells. Gross. Spirochetes are helical heterotrophs. Syphilis, and then Lyme disease is probably the one that more people are familiar with. Cyanobacteria are able to make oxygen. And then gram-positive bacteria that we've got anthrax, Clostridium botulinum. So you'll hear of, I believe that is a source of how they're able to do Botox, but I was thinking of botulinum. There's a poisoning, but I'm not coming up with it right now, and I'm trying not to drag this out. Staphylococcus, streptococcus, strep throat. Those are other sources of common bacterial infections. And then mycoplasms are super, super tiny and can cause all sorts of trouble. So those are examples of your different bacteria. Be fun. Prokaryotes. If we did not have these, there would be some pretty significant issues. They help to recycle things. They help to break down things. So they're able to decompose, they're able to recycle, and they help plants get the nutrients that they need to be able to grow. But they can also cut down on the availability of those nutrients. So again, there's pros to prokaryotes, but there's definitely a lot of cons. We'll spend more time with these ecological interactions in our last unit. You can have species interact with species that live close to one another. Sometimes they, and that type of relationships, when they work in or live in live in contact closely with one another, is called symbiosis. Sometimes those symbiotic relationships are positive, where they're mutualism, both species get something from it. Sometimes they're commensalism, where one species gets benefits, the other one isn't harmed or benefited by that relationship. And then we have ones that are parasitism, where you don't kill your host, but you definitely harm it. And when you cause diseases, those are called pathogens. It's part of why you treat your pets for fleas. It's part of why dogs are treated for heartworms. Parasites are not so good. So again, we'll spend more time on that with ecology. So again, we talked a little bit about the pros and cons of prokaryotes. Mutualistic bacteria, our intestines have lots of different bacteria, and they help to break down the food that is in our intestines that does not get digested. And then we have bacteria that can harm us. We talked about Lyme disease. I definitely pointed out some others that were there for you as well. They can do this when they cause diseases by releasing exotoxins or endotoxins. The exotoxins are released and they can cause disease even if the prokaryotes aren't there anymore. The endotoxins are released only when they die. So they're housed within the bacteria, and when they die off is when the endotoxins come out. And then we can have the gene transfer that can make things a little bit more of an issue. And there's the issues with bioterrorism and fun stuff. Okay. Um, we've been able to use prokaryotes to get further along with DNA technology. Again, we can use E. coli with gene cloning. That was what we used when we were doing our transformation lab. We can use some bacteria, Agrobacterium tumefaciens, to make plants that have taken up DNA, transgenic plants. We can have bacteria with those plasmids inserting genes to be able to make natural plastics. Bioremediation, we can help them help remove things from the environment. Um, we can have them make vitamins and antibiotics and hormones. We can help them to make ethanol. So there definitely are some benefits, but it has to be done with caution.